and prevention activity specifically and conducting the cognitive aspects, including risk factors, renal injury, mechanical, and drug compounds. And today, Professor Lian will give us a talk with the title Unlocking the Door of Cellular Climate Improvement, the key driver in renal fibrosis. So let's give our welcome to Professor Lian.
First of all, thanks all the members for invitation and giving me this great opportunity to present our work here. So, today I'm talking about the story of cellular iron protein. So, iron protein is famous for the map for disease, right? So, but today I'm talking about his story in the pathogenesis of kidney disease. Okay, so chronic kidney disease actually is a major public health problem with high morbidity and mortality worldwide. And kidney is also the third largest growing cause of death worldwide. Unfortunately, so far there's no targeted therapeutic agents or strategies specifically therefore so. And dissecting underlying mechanism is still challenging and very important for developing new diagnostic and therapeutic. So when thinking about this chronic kidney disease, actually, people would like to think that CKD is kind of a premature aging syndrome. So from our patients' clinical symptoms, we can see the patient has osteoporosis, myocardial hypertrophy, cognitive impairment, and vascular carcinogen. And one of the changes of the chronic kidney disease is the renal fibrosis, which is a degenerative pathological change that occurs in many organs. And mechanistically, cellular senescence is also a key driver for the pathogenesis and also the progression of kidney disease. So, therefore, when we talk about aging-related disease, and we all know that neurodegenerative disease is the most expensive chronic aging-related disease. Therefore, we're thinking that is it possible that the CKD and neurodegenerative disease share some common mechanisms? So, a key hallmark event of neurodegenerative disease is the accumulation of misfolded protein activation, such as amyloid oligomerization detected in Alzheimer's disease, and also the alpha-synuclein form So whether the protein misfolding and activation contributes to the pathogenesis of renal fibrosis, which is remains largely unknown. So we started to search the published signatures for those misfolded protein, and we found a very interesting finding is that the plasma level of cellular iron protein is significantly activated in patients with renal failure, and also its concentration in the patient's uremia is significantly elevated and closely correlated with the decline of the renal function of the patient. So this gives me the question is whether the cellular iron involved in the pathogenesis of CKD or just a correlation reason. So to do that, to answer these questions, we generated two classical mouse biological models. So one is the EU model and the other is tubular replication model. So we can see that from here that both the messenger RNA and also the protein level of PRPC are dramatically activated in the fibrotic kidney tissues. And research accomplished single-nuclear RNA sequencing data confirmed or founded is that, first of all, cellular iron expression is upregulated, and in most accumulate in the proximal tubules and also in the active fibroblasts. However, when we do the immunofluorescence, we can only confirm the co-localization of the fibroblasts in the proximal tubules, but not in the active fibroblasts. And then we turn to our patient samples. Here are the renal biopsy CKD patients. So we can see dramatic activation of fibrin protein in the cardiac and the predominant renal region of our CKD patients, regardless of this etiology. And also the expression level of fibrin is correlated with the time of the renal function and also the severity of the renal fibrosis. So here is the published spatial transcriptional data and confirm our finding that the cellular protein is accumulated in the proximal tubule cells of our CKD patients. So therefore, we generated the tubule-specific fibrin knockout mice and also to subject them to the new model, the fibrosis model, and you can see that knocking out the fibrin could mitigate the fibrosis. This is another model, the skin reproducing model, we also see mitigation. So the next question is, what's the mechanism by which the cellular fibrin help to promote the fibrosis? So to address this issue, we collected the RNA with all RNA sequencing of the cells overexpressing cellular fibrin. And from this data, you can see that 70% of the upregulated gene in the fibrin overexpressing cells are the target gene of integrin regulatory family, factor families. So therefore, we confirm the distribution of these phosphate these cells. So we see the aggregated phosphorylated TDP1, aggregated phosphorylated IL-3, which is our expressing cells, and also in our UO models. And then we also examine expression of phosphorylated TDP1 in our CKD patients with biopsy. We see the co-localization of cellular fibrin with the phosphorylated TDP1 in the same renal tubules. And also the level of phosphorylated TDP1 in the renal tissue is correlated with the patient's renal dysfunction. So therefore, we conclude that the cellular fibrin could activate TDP1 RF signaling in the fibrotic renal regions. So next question is that we look at to find the target gene of the RF family. So the genome analysis tell us that fibrosis-related genes are enriched in the cells. So in the target gene of IRF, such as bacteria, chemofunction, and extracellular matrix monopolization, etc. And also we see that the fibrosis-related genes like alpha-synuclein muscle-actin and fibronectin are the direct target of IL-3. So we do the chip assay to confirm that IL-3 indeed comes from the hormone region of alpha-synuclein and fibronectin, and this finding is elevated in cells overexpressing cellular fibrin. So the last question is how does fibrin activate this contact, this single pathway? So previous studies have shown that the N-terminus of the fibrin contains intrinsically disordered region, so which is the characteristic of cells which is a protein that has capacity for liquid phase separation. And most recent study also revealed the C-terminal of fibrin also to undergoes the LFS. So what we do is we do mutant a couple of key amino acids in the fibrin sequencing. So from this data we can see that when we transfect the well-tapped fibrins into the new tube, this is the real tubule cells, we can see those condensates localized in the cytosol of the wild type in the cytosol, in the renal tubule cells. However, in the lysine to alanine mutants, K2A mutants, we fail to see the common cells in the cytosol. Similarly, so when we also this mutant fail to activate the TDK1, IL-15, and also fail to upregulate the fibronectin and allosynuclein actin. So this is the major kinase assay. So we can see that when we add one factor of PRP in the literature, we see that upregulation of phosphorylated TDK1 and phosphorylated IL-3 are relatively limited. So since we have observed that the TDK1 IL-3 treatment is important to drive the fibrosis in it, then we see that whether we could, if we block this signaling pathway, we mitigate the fibrosis. So anaximox is FDA approved TDK1 inhibitor to treat asthma for patients. So that's why we implement the anaximox in this high model for 3 days. And we can see the mitigation of the fibrosis in this model. So the next thing we do is that we give the mouse to a model that with the established renal fibrosis. So which means that we give the mice the adenine to generate the chronic CKD model and by injecting the mice with adenine. So after 28 injections, we can see the obvious fibrosis lesion in the kidney, and then we start to give the TDK1 inhibitor at this time point for another 2 months. And we see that very dramatically attenuation of established renal fibrosis by this treatment. So here is the take-home message. So what we found is that we found that the cellular firing is activated in the fibrotic renal region and within of our CKD patients. So the level of the renal firing is correlated with the severity of the renal fibrosis and the decline of renal function in CKD patients. And cellular firing activates the TDK1 IL-6 pathway through its capacity for base separation and promotes the pro-fibrotic response in renal tissue cells. Pharmacological inhibition of the TDK1 IL-6 pathway to block PRC-induced pro-fibrotic response, mitigate renal fibrosis, increase renal function in preclinical CKD animal models. So I'd like to thank the 4 graduate students have finished this fantastic work and thank